Protein synthesis or what is called gene expression بتضمن يعني تصنيع البروتين يتضمن عمليتين. أول عملية transcription والعملية الثانية اللي هي الترانسليشن. الترانسكريبشن معناته تصنيع ماسنجر RNA من الدي ان اي، والعملية الترانسليشن ترجمة الشفرة تبعت الماسنجر RNA لبروتينات. خلينا نشوف الترانسكريبشن. It is the synthesis of RNA under the direction of DNA. الماسنجر RNA هو اللي بيتصنع من الدي ان اي، يعني واحدة من التو ستراندز تبعت الدي ان اي راح يصير لها ريبليكيشن واحدة بس. هلا الستراند اللي انتسخت يعني الستراند الجديدة هي الماسنجر RNA. طيب وين بتتم العملية الترانسكريبشن؟ بتتم في النيوكليوس، لأنه الدي ان اي موجود في النيوكليوس في النواة. RNA synthesis or transcription is the process of transcribing DNA nucleotide sequence information into RNA sequence information. كأني بدي أخذ الشفرة تبعت الدي ان اي وأحطها في الماسنجر RNA. DNA sequence is enzymatically coupled by RNA polymerase to produce a complementary nucleotide RNA strand called messenger RNA because it carries genetic message from the DNA to the protein synthesizing machinery of the cell. طيب لما أصنع messenger RNA من الدي ان اي معنى الكلام يعني الترانسكريبشن هو تصنيع الماسنجر RNA من الدي ان اي. طيب لما أصنع أنا ستراند من الماسنجر RNA من الدي ان اي، الدي ان اي يحتوي على ثايمين ويحتوي على دي اوكسي رايبوز، ففي شغلتين لازم أغيرهم في الكبي اللي بدي أعملها من الماسنجر RNA، إنه أحول الدي اوكسي رايبوز لرايبوز، وبنفس الوقت أحول الثايمين إلى يوراسيل يعني بالتخلص من CH3 اللي حكينا عنها. هنا اللي بيصير في الترانسكريبشن، هاي تبعت الدي ان اي اللي لونها أخضر، ويتم تكوين كومبلمنتري ستراند لهاي الستراند، وبتكون سلسلة واحدة من الماسنجر RNA هي اللي مسؤولة عن الترانسكريبشن. طيب لاحظوا الماسنجر RNA كيف عبارة عن قواعد نيتروجينية، كل 3 قواعد نيتروجينية مع بعض بنسميهم كودون، فيحمل الماسنجر RNA مسج اسمها كودونات. تمام؟ طيب هلا DNA template strand is read من 3 لل 5 برايم by RNA polymerase, and the new strand RNA strand is synthesized in the 5 prime 3 prime direction.
In today's video, we're going to take a closer look at protein synthesis, which is the process of making proteins. We can think of this in terms of 2 steps: transcription and translation. In a nutshell, transcription is the process of taking a single gene of DNA and copying it into a structure called mRNA. Then translation is the process of taking this mRNA strand and using it to produce a protein. Before we go through the details though, we need to look at why we actually need these 2 steps. Inside almost every cell is a nucleus that contains all the genetic material of that cell in the form of DNA. The reason we say DNA is so essential to life and controls what the cells do is because it contains thousands of genes, which are each small sections of the DNA that have a specific sequence of bases, and so are able to code for a specific sequence of amino acids, which when combined will form a particular protein. In order to actually make a protein though, the specific sequence of bases has to be read by one of these structures, called ribosomes, which importantly are outside of the nucleus. However, because the DNA is so big, it can't leave the nucleus itself. And so if we want to use a gene to make a protein, we're going to have to make a copy of that gene first. And because we're only copying a single gene, rather than the entire DNA strand, the copy will be small enough to leave the nucleus, and so it can make its way to the ribosome. Now, this copy we're talking about is mRNA, which stands for messenger RNA, and is just a copy of a single gene. The structure of mRNA is mostly similar to DNA, but it does have a few important differences that you need to know about. For one, it's much shorter than DNA, because it's only a single gene long. It's also only a single strand, rather than a double strand like DNA. And finally, instead of containing the base thymine, it contains uracil. Now that we've got these basics covered, let's take a closer look at transcription and see how this mRNA is actually formed in the first place. In this image here, we can see a small section of DNA that contains two strands coiled into a helix. This is how it's normally found in the nucleus. But to make it easier to understand transcription, let's uncoil the two strands and show it using this simplified diagram instead. To be clear though, this is the same piece of DNA as above. We're just showing the two strands side by side, rather than wrapped around each other. And we're showing the bases as their letters. So A for adenine, T for thymine, and so on. For the sake of our example, let's say that this region here, between these two lines, is the gene that we want to copy. Even though in real life, genes are normally much bigger than this. The process starts with an enzyme called RNA polymerase, which binds to the DNA just before where the gene starts. Then, just ahead of the RNA polymerase, the two strands of DNA separate apart, so that all of their bases are exposed. Then the RNA polymerase is basically going to move along the DNA strand and read the bases one by one and use them to make an mRNA strand. For this to make sense though, you need to remember that the mRNA bases will always be complementary to the DNA bases. So a C on DNA will always pair with a G on mRNA, G will pair with C, T with A, and then the odd one out is that if you have an A on the DNA strand, it's going to pair with a U on the mRNA strand, because mRNA doesn't have T. Like we said a minute ago, all of the thymines, which are the T's, have been replaced with the uracils, which are U's in mRNA. So an A on DNA will be complementary to a U on mRNA. So in our example here, the RNA polymerase will start with this C base. And so it will start the mRNA strand with a complementary G base. Next, it will read the T base. So it adds a complementary A to the mRNA. Then it will move on to this A base and add a complementary U. Then go to the G and add the complementary C, and then so on. So the RNA polymerase is going to carry on doing this, building up the mRNA base by base as it moves along the entire gene. One thing to notice here is that the DNA strand keeps on separating just ahead of the RNA polymerase and closing just behind it, so that only a small section of the DNA is ever exposed. Once it's moved along the entire gene and finished making the mRNA strand, the RNA polymerase detaches from the DNA, and the DNA strands can close back up. This means we're left with an mRNA that's then free to leave the nucleus and head off to the ribosome. It's called the template strand. So it's the template strand which is used to make the mRNA.
طيب، نتيجة عملية الترانسكريبشن يتم تصنيع المسنجر RNA من الدي ان اي في النواة. ليش؟ لأنه الدي ان اي كبير، ما بيقدر يطلع برا النواة، هو عايش في النواة، مملكته في النواة، فما بيقدر يبعث للريبوزومات اللي موجودة في السيتوبلازم رسالة إلا عن طريق تصنيع messenger RNA، والمسنجر RNA هو الرسالة، بيحمل الرسالة. طيب نيجي لقصة الترانسليشن اللي هي تحويل المسج اللي جايبها المسنجر RNA إلى بروتين. كيف؟ Translation is the first stage of protein synthesis. Translation is the production of protein by decoding mRNA produced in transcription. يعني حل شفرة المسنجر RNA يؤدي إلى إنتاج البروتين في الريبوزومات. Translation occurs in the cytoplasm where the ribosomes are located. This process uses a messenger RNA sequence as a template to guide the synthesis of a chain of amino acids that form a protein. The messenger RNA carries genetic information encoded as a ribonucleotide sequence from the chromosomes to the ribosomes. يعني المسنجر RNA يحمل ما يعرف بنيوكليوتايد تريبلتس، يعني مثلا AAU GGC CCA. هدول بنسميهم الكودونات. الكودون هو الشفرة اللي لازم تنقرأ، والشفرة تدل على أنواع الأحماض الأمينية اللي يجب إضافتها لحتى نعمل بروتين. تمام؟ Each of those triplets codes for a specific amino acid. طيب لاحظوا مثلا هاي التريبلت UUA UUU معناته أنا في عندي فينيل ألانين، UCU لازم سيرين، UAU تايروسين، وهكذا. هدول الكودونات اللي لازم، يعني كل كودون يدل على حمض أميني معين. تمام؟ لما بيكون الكودون هذا موجود على المسنجر RNA وبينقرأ على طول بيقول إنه أضف فينيل ألانين، UCU أضف سيرين، UAU أضف تايروسين، وهكذا. طيب هل يستطيع المسنجر RNA إنه يعمل ترانسليشن لوحده؟ أبداً، محتاج ل3 شغلات. واحد، الريبوزومات إلها قطعة كبيرة وقطعة صغيرة، بيكونوا عادة بعاد عن بعض. لما يجي المسنجر RNA على الريبوزومز بيقربوا على بعض. مين بيخليهم يقربوا عادة؟ الريبوزومال RNA اللي هو rRNA. معنى الكلام إنه في عندي ريبوزومز وفي عندي rRNA. طيب مين الثالث؟ التي RNA اللي هو ترانسفير RNA. شو بيعمل ترانسفير RNA؟ هو عبارة عن سيارة. هاي السيارة حاملة جواها حمض أميني معين، ومش بس هيك، في إلها إيد. هاي الإيد اسمها أنتي كودون، كأنها مفتاح، وبيجي المسنجر RNA هو قفل بالكودونات تبعته. يعني كل كودون عبارة عن قفل، بيجي التي RNA مع المفتاح. إذا فتح معناته بيخلي الحمض الأميني اللي داخله ينزل. وين ينزل؟ عند الريبوزومات. يقول لهم يلا خدوا هاي أول أمينو أسيد ضيفوه. طيب بيجي التي RNA مرة تانية على القفل الثاني، بيحاول بيحاول بيحاول، أي تي RNA بيزبط بيفتح القفل تبع المسنجر RNA، بيقدر إنه الأنتي كودون يطابق الكودون، معناته بيقول للحمض الأميني اللي معه ينزل. وهكذا يتم إضافة أحماض أمينية واحد وراء الثاني على داخل الريبوزومات أو باستعانة بالريبوزومات، وتكوين سلسلة من الأحماض الأمينية اللي هي البروتين المطلوب. فبنلاقي إنه الماسنجر RNA مع الريبوزومال RNA مع الريبوزومات مع التي RNA، الأربعة هدول تساعدوا لتصنيع البروتين. هذا الحكي في السيتوبلازم، وبدون ما يتصنع المسنجر RNA في الترانسكريبشن في النواة ما بيصير هذا الحكي كله، لأنه اللي بده يبلش عملية الترانسليشن هو وصول المسنجر RNA للسيتوبلازم للريبوزومات. الشكل اللي احنا شايفينه هون هو شكل مين؟ التي RNA. وبيهمني نعرف منطقتين فيه، منطقة حاملة للأمينو أسيد أو ماسكة بالأمينو أسيد، معنى الكلام إنه هذا التي ار ان اي حامل وناقل لهذا الحمض الأميني اللي هو اليوسين واحد بس. ومنطقة ثانية اسمها أنتي كودون، إلها 3 كودونات، إلها 3 قواعد نيتروجينية اللي هما أنتي كودون. هلا شو بدهم يقرأوا الأنتي كودون هدول؟ بدهم يقرأوا المسج تبعت الار ان المسنجر ار ان اي اللي هي كودونات. لاحظوا إنه الجي لازم يربط مع السي، الاي مع اليو والاي مع اليو. إذا ما لقوا هذا الكودون ما بيقدروا يربطوا معه، فلازم يكون مطابق الأنتي كودون للكودون. هون الحمض الأميني هون بيترك وبينضم للريبوزومات. طيب خلينا نشوف السمري اوف بروتين سينثيسيس. Gene expression requires 2 steps: transcription, translation. Messenger RNA transcripts are synthesized, modified in nucleus من ال DNA. Messenger RNA is moved to ribosomes in cytoplasm. Translation occurs on ribosomes. Many ribosomes can translate the messenger RNA transcript simultaneously. First few amino acids of polypeptide signal peptide that determines where in the cell the polypeptide belongs. Polypeptides enter the lumen of the endoplasmic reticulum to be folded and processed. Transport vesicles carry the protein between organelles and to the membrane. يعني في خطوة أخيرة بعد تصنيع البروتينات إنها تروح على الاندوبلازميك ريتيكولم لحتى يصير لها فولدينج، يصير لها conformation معين، فبالتالي تحمل وظيفة معينة.
A copy of the gene here inside the nucleus, which is free to leave the nucleus and make its way to the ribosome, where it can undergo translation to produce a protein. To help understand this part, it's important to remember that for both DNA and mRNA, each group of 3 bases, which is called a triplet or codon, codes for a specific amino acid. To make proteins, our cells use 20 different amino acids, and each one has a different 3-base codon. For example, this triplet, AGU, codes for the amino acid serine, whilst CCA here codes for proline. You don't have to remember these examples. We're just using them to illustrate the point. So let's now zoom in to a single ribosome and go through the process of translation. To start the process off, our mRNA strand and the ribosome both bind together. And we're now ready for the ribosome to start building the protein by adding 1 amino acid at a time. The amino acids themselves are brought to the ribosome by molecules called tRNA, which stands for transfer RNA. tRNA molecules have the amino acid at the top and an anticodon at the bottom. The anticodon is this sequence of 3 bases, which are complementary to these 3 bases on the mRNA. And it's these 3 bases on the mRNA that code for the amino acid that the tRNA is carrying. So because each type of tRNA molecule is specific to a particular triplet on the mRNA, it can ensure that it always brings down the correct amino acid. For example, because the first triplet of our mRNA sequence here is AGU, it will attract this tRNA molecule with the anticodon UCA, because UCA is complementary to AGU. And tRNA brings with it the amino acid serine, because AGU is the code for serine. Meanwhile, this second triplet, CCA, will attract this tRNA molecule with the complementary anticodon of GGU, and that will be carrying the amino acid proline, because CCA codes for proline. Now, the whole point of this seemingly complex process is that the tRNAs have now brought the correct amino acids down to the ribosome in the correct order. So the ribosome is able to join them together and start building up a chain of amino acids. Once it's joined these first 2 amino acids together, the ribosome moves along the mRNA slightly, and so another tRNA molecule will come down and bind to its complementary codon on the mRNA. bringing with it the next amino acid. This allows the first tRNA molecule to detach and repeat the process. But importantly, it leaves the amino acid behind. This same process then repeats all the way along the chain, until the ribosome reaches the very end and has joined together a complete chain of amino acids. Reaches the very end and has joined together a complete chain of amino acids. At this point, the amino acid chain will detach from the ribosome. And then finally, the chain can fold up on itself to form a protein.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain.
لاحظوا هون إنه إحنا في عنا ماتش بين الأنتيكودون على التي ار ان اي اللي هو سي يو اي والجي اي يو اللي هو الكودونات على المسنجر ار ان اي. هذا اللي 5 برايم 3 برايم هو مسنجر ار ان اي، فلازم يصير في ماتش، يعني الجي لازم تربط مع السي والاي مع اليو واليو مع الاي. هيك في ماتش بين الانتيكودون على التي ار ان اي والكودون على المسنجر ار ان اي. في هاي الحاله لما بيصير في ماتش التي ار ان اي بيترك الحمض الاميني وبيطلع، بيدخل من جهه عاده وبيطلع، بس بيدخل معاه الحمض الاميني، ولما بيقرا الانتيكودون بيقرا الكودون بيطلع بدون الحمض الاميني، بيروح يجيب نفس الحمض الاميني اللي بيقدر يحمله هو او بيقدر ينقله.
Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.